Good afternoon. Welcome to the Wednesday afternoon lecture series. I'm Marguerite Matthews, a program director at the National Institute of Neurological Disorders and Stroke. On behalf of NINDS and our sibling neuroscience institutes, I am thrilled to introduce today's WALS speaker, Dr. Kafui Dzirasa, who has pioneered groundbreaking research at the intersection of neuroscience, psychiatry, and engineering. Before I get into his very impressive resume, I have two very quick announcements. The first is the code for continuing medical education credits. The code is 57693. I'll repeat that, 57693. For those of you watching remotely, you can submit questions to the speaker anytime during the lecture. Simply click on the button that says Send Live Feedback, which is below your video cast window. Type in your message and we'll relay it to the speaker at the end of the lecture. Now to our guest of honor. Kof, as he is affectionately known, is an A. Eugene and Marie Washington Presidential Distinguished Professor and Howard Hughes Investigator at Duke University School of Medicine. His research spans a wide range of disciplines, as evidenced by his many other titles at Duke. These include Professor of Psychiatry and Behavioral Sciences, Professor in Neurosurgery, Professor in Neurobiology, Assistant Professor of Biomedical Engineering, and Investigator in the Duke Institute for Brain Sciences. Dang, man, your signature must be long. At his core, he is a psychiatrist and neurobiologist with an interest in discovering how emotions are encoded in the brain in health and disease. The essence of his research is the study of how neuropsychiatric risk genes interact with environmental stress to modify neural circuits that underlie normal emotional and cognitive function. His lab uses in vivo electrophysiology, cell type-specific neuromodulatory techniques such as optogenetics, and quantitative analysis of behavior in genetically modified mice. They were the first to identify patterns of electrical activity across multiple brain regions, known as network-level oscillatory dynamics, that predict vulnerability of depression-like behavior in animal models. This work has advanced our understanding of mood disorders and opened new avenues for circuit-based therapeutic interventions. Kof earned a BS in chemical engineering from the University of Maryland, Baltimore County, right up the road. All right, okay. UMBC in the house. And then he went on to receive both his PhD in neurobiology and MD from Duke University School of Medicine, where our paths first crossed. Kof also completed a psychiatry residency and postdoctoral training at Duke. Beyond his academic and research achievements, Kof served on the advisory committee of the director of the NIH for 3 years and has received numerous honors, including the NIH Pioneer Award in 2022, and has been elected to the National Academy of Medicine in 2021. He is also a passionate advocate for broadening participation in neuroscience through mentorship, outreach, and policy engagement. The title of his lecture today is Towards Preventative Psychotherapeutics. Please welcome Kof.
Well, it's a real honor and a pleasure to be. I call this my second home. I grew up in Silver Spring, spent some time here on NIH's campus as a medical student, and have been back multiple times since with friends and colleagues across the institution. So I'm going to give a talk today about a problem that I've been trying to solve for the last 20 years. And when Marguerite was giving my introduction, she sort of mentioned all the different departments I was in and associated with. It is because I've reached into all of those disciplines to try to solve this problem. And I haven't done that because it's been fun. I've done that because it's been necessary to try to make headway in solving this problem. So the trainees here, I always like to give the disclaimer for trainees, which is you should not give a talk like this, right? We're going to course and zip through all kinds of disciplines. I'll do my best as I can to orient you as we go in and out of those spaces, but it is to solve a problem that has turned out to be way harder than I thought it would. All right. So I always like to begin with this slide to point out how much burden of illness, this is years of productivity loss, we experience from mental illness. And in terms of global loss of productivity, so this global burden of illness, mental illness is on the top of the list. I used to take 4 or 5 slides to walk people through this topic. I think after the pandemic, we all appreciate how deeply impactful these illnesses are. We've seen them in ourselves and our family members and our colleagues. And I just wanted to show this data. It's not just the pandemic. This is youth. You can see the same thing with anxiety features. The incidence and the prevalence of these disorders have been going up over time. They certainly went up with the pandemic, but they were increasing as well. So a little bit of my background for trying to think about how to get traction on these illnesses. I always thought a really good starting point was understanding the biology, and the genetics would provide us some really good clues. So as I was coming up in medical school and graduate school, we launched these GWAS studies, these genome-wide association studies. And the idea was that we would sequence the genome, right? And then we would find hits that give risk for mental illness, right? So we'd find the gene for depression, the gene for schizophrenia, in the same way that we sort of found the gene pathways for cystic fibrosis. And once we found those, we can ultimately fix them and fix illness. It turns out, as we were doing this, the landscape for psychiatric illnesses were way more complex. There were lots of different risk genes that could come together to form illness. And over time, what happened was we started making our studies larger and larger and larger and finding more and more and more hits. And so as a field, we wanted to make sense of this. And what we came to appreciate over time was that many of these sort of biological risk factors seem to be attaching or converging on a space at which brain cells interact called the synapse. So this connectivity space between brain cells. So I'm going to show you an image here, just sort of summing up some of the findings. In the yellow color here and the red color, the red identifies sort of genes and proteins that have been implicated in schizophrenia. The yellow in autism spectrum disorder and intellectual disability. You could see that these hits were not like just one illness. They were showing up across illnesses that impact psychiatric and emotional function, right? But they were converging on this space called the synapse. It just wasn't one protein. It was many different proteins. But that's where the idea for how we could think about intervening on these came up. As Marguerite mentioned, I'm a biomedical engineer. So I wondered if we could sort of use technology, if there's like a disruption in this sort of communication pathway, could we basically use technology to go around it, right? If your landline for your telephone is messed up, can we essentially create a cell phone that goes around the disruption? So imagine you have these 2 cells. On the top, you see the presynaptic cells. Let me see if I can use this pointer. Yeah, it works. You have presynaptic cells, and then you have postsynaptic cells. So imagine you have a blockade. Oh, well, there's that too. Okay, well, you probably get it from the picture. The red X is the blockade. So you have a blockade, and so the question is, could you essentially go around the blockade? Can we bypass it? So could we have a technology that reads information from the presynaptic cell and passes it to the postsynaptic cell? And this isn't some great, grand, big idea. This is something that I saw in the clinic. And so this is a heart, and the heart has to be supplied by blood. And there could be a set of conditions in which there's a blockade in the heart. And then the blood vessels, blood can't get from where you're sending it to where you want to receive it in the muscle tissue, right? And there's a technique that was created. We've probably heard about this, quite ubiquitous, a coronary artery bypass graft. And what you do in the bypass is you basically put a blood vessel in front of the blockade, and then you attach it after the blockade, and blood can flow around the blockade. So pretty simple conceptually. And so we had an idea of essentially creating a neural bypass that bypasses a synapse. And here's what our first design would look like. We'd use a set of tools where we had our presynaptic cellular population, and we're going to record electrical activity. So we're going to just implant an electrode in the brain and sample electrical activity upstream. And then we were going to feed that information in the computer and sort of figure out what it was supposed to be saying. And then we would play that information downstream after the synapse. And the way we play that information into the brain is we'd use a technique called optogenetics. Conceptually, it sort of works the same way your eye works. It changes light into chemical energy. Here, the light is changed directly into electrical energy because a channel opens up and closes. And so light equals stimulation of brain cells. And so we're going to record information, process it in a computer, and then drive it back into the brain. And we've got to do this with sort of a 20 millisecond delay, so 1/50th of a second. And so an undergrad working in my lab, just to give you an indication of how long this has taken, he's now applying for faculty positions. An undergrad working in my lab, we came up with this schematic to essentially create this sort of technology. So we're going to record information from the brain, process it in a computer, and then feed it back in with some high-frequency pulses. And the way channel rhodopsin works is the channels open up when you stimulate with blue light. So blue light is the experimental group. Yellow light is the negative control. In other words, yellow light does not make the brain cells fire. And we wanted to pick a paradigm in which we can test this type of stimulation device in. And my lab is really interested in stress biology, so we wanted to pick a context associated with stress behavior. And I always put this picture up, and I like to explain it. I'm a psychiatrist, right? People have used this as a model of depression. The mouse is not depressed. We don't do this to humans to measure depression. There are only two things I need you to accept so we can move forward in how we then tested out this technology. The first thing that one must accept is that this context is stressful for the mice. We can objectively determine that because we can measure the mouse's stress hormones. So you do this to a mouse, its stress hormones go up. The second thing is the mouse's behavior in this assay is really sensitive to how much stress it has previously received. So if you stress the animal out and put it in this assay, the animal sort of wiggles around less. So the assay is stressful, and the assay is sensitive to the animal's prior stress. So we took advantage of this assay by essentially running it 2 days in a row. So you're measuring the animal's responsiveness to stress on the first day and the second day, but on the second day, it has previously been exposed to the stressor of the first day. And that allows us to ask the question, how does its computation of behavior change in the same context, with the same context occurring between day 1 and day 2, but on the second day, it's been stressed on the previous day. So when you look at animals behaviorally in this assay between day 1 and day 2, they're not all identical, right? But as a population, they tend to show more immobility on the second day than on the first day. So that's how they adapt to the stress exposure. So what we're going to do here is search for which sort of synapse, which circuit connection we should be targeting for the intervention. So we did this by implanting electrodes into the brains of mice, and then once they recover, we're able to record electrical activity from many brain cell areas, brain regions, at the same time. And in this assay, we're going to be recording from a set of regions, basolateral amygdala, dorsal hippocampus, infralimbic cortex, prelimbic cortex, medial dorsal thalamus, nucleus accumbens, core and shell, and the ventral tegmental area. And it's not quite the power of a technique of an fMRI, a functional magnetic resonance imaging that's looking at the whole brain, but we're getting a good survey of lots of brain regions that have been involved in stress computation. And then we're going to ask which of these brain regions show changes in their computation between the first day and the second day, right? So when we do that, and we're serving these brain regions, there's only two brain regions that change the way that they're encoding. The first one is infralimbic cortex, so a part of what we would call mouse prefrontal cortex. And the second one is medial dorsal thalamus. I'm just showing you the cells here and how the computation is changing. It only identified two brain areas that were coding differently. The other brain areas were coding fine between day 1 and day 2, day 1 in gray, day 2 in red. So we then focused on these interconnected brain regions for the rest of our study. And what we wanted to understand was how encoding or information processing was happening between these areas. We searched through a bunch of different ways that brain areas can sort of communicate or synchronize with each other, and we found this really unique pattern between infralimbic cortex and medial dorsal thalamus. And I'll sort of use music to explain what's going on here. So this pattern was called cross-frequency coupling, right? And so you can think of things being synchronized. Things can be synchronized like this. Things can be synchronized like this. Or things can be synchronized across frequencies like this. So we tested that pattern, and we found something that looked like this, with the green being the lower frequency oscillation, and the red being the higher frequency oscillation. And you could see that the amplitude waves of the red oscillation tend to happen at a part of the cycle of the green oscillation. So that's called cross-frequency coupling. Here's another way of looking at that type of plot. You could see on the y-axis, we have the amplitude of the oscillation of the red. On the x-axis, we have the phase of the oscillation of the green. Blue means no coupling. Red means high coupling. So we basically see coupling between these two bands of frequencies, the sort of 2 to 7 hertz of infralimbic cortex and the 30 to about 60 of medial dorsal thalamus. And when we look at that type of coupling between these brain areas, that type of coupling changes between day 1 and day 2. It gets much stronger on the second day than on the first day between these two brain areas. So as an engineer, we had a question. We thought, well, maybe this is the stress change that matters, right? And if this is the stress change that matters, we want to figure out how to get rid of this, and we'll make the animals more resilient to stressors, right? On the other hand, maybe this signature is compensatory, right? And this is always something we have to think about as engineers, right? If I run up a flight of stairs, my heart starts going faster because it's a compensation, right? And so if we have compensation, what I don't want to do is my heart not to go faster when I'm running up a flight of stairs, or I won't be able to run up another flight of stairs, right? Because the heart is compensating to keep my muscles oxygenated. And so we have to figure out if this is primary, this is the problem, or this is compensatory, because that dictates how we intervene on it for the sake of health. So what we're going to do is, to figure this out, we're just going to play this signature back in the brain, right? We're going to read information from infralimbic cortex, and we're going to play it in the thalamus. And if it is primary, it'll make the animal's behavior worse. If it's compensatory, it'll make their behavior better. So here's what the data looks like. You can see it on the left. The animals, we've got them on the assay. Blue light is our experimental group. Yellow light is our control. And as you can see, the blue group is less immobile. In other words, playing the signature into their brain reversed the phenotype. In other words, it is a compensatory signature. It is not primary. It is compensatory. And so by playing this compensatory in, we increase the animal's resilience to an external stressor. In other words, strengthening this pathway. We did a series of controls, which we also thought were really important, and these were really instructive. You could see on the right, we're basically stimulating time to the oscillation in infralimbic cortex. In other words, we're doing our read-write, we're doing our bypass. For our first control, we said, well, maybe it's just because we're stimulating thalamus. So let's take the same number of light pulses, but let's just stimulate it in a way that isn't restoring the timing in this circuit. So we're just going to stimulate with a normal type of pattern that people use all the time in the animal research literature, same number of light pulses, and it actually made the animal's behavior worse, right? So the first one made it better, the second one made it worse. It's not just the cells. And then we thought of another control in which we said, well, maybe it's the burst of 3 that are really important. So we stimulated with the burst of 3, and we got a pattern that looked like this, right? The behavior didn't change at all. And we think what's happening is it's actually an issue that engineers can appreciate. It's called signal to noise, right? So in the first case, you time the signal correctly, you've increased the signal. In the second case, you time it incorrectly, you've increased the noise. And in the third case, some of the 3 pulses are at the right phase of the oscillation, and some are at the wrong phase. So you've increased the signal and the noise, and so nothing happens. So what this means is if we ultimately want to think about doing the synaptic bypass, we have to get an entire system that looks like this that reads the information and plays the information back in with the right timing. So this was going to be the solution. I set out to solve a major problem, and we were excited because we'd finished and figured out the solution to this major problem that we were trying to solve. And I was so excited that I went back to my home. My family is from Ghana and West Africa. And my motivation for this work that I do, the bottom row is my mom and her siblings. And 4 out of 5 of them carries a major psychiatric diagnosis, whether schizoaffective disorder, bipolar disorder, or depression. And so my motivation towards coming up with therapeutic devices it's an extremely selfish one. It's like not curiosity. It is love for my family members and wanting to heal them and heal the next generation of my family. And the open chair, there's my uncle who has bipolar disorder. This is a picture from a grandmother's funeral. And the stress of that situation had caused him to decompensate, so he was unable to make it. And the rest of this talk is going to be motivated by a conversation I had with him, in which I was really excited. I showed him this design for this bypass mechanism. I was explaining to him. And he said, this is really good. The family's so proud of you. We feel like our generation of illnesses reached you, so you would do something good for the world. Tell me more about how this is going to get to the third world. And me as an engineer who thinks about applications and impact had never thought about the fact that what I was creating was actually never going to get to the third world. It was never going to scale. It was always going to require neurosurgeons, of which are not enough available in technology. And the global burden of illness, of depression and bipolar disorder and schizophrenia and addiction is so large that unless you had a technique that was scalable, it actually wasn't going to move the needle in the burden of illness. So this took us back to the drawing board, and the remainder of my talk will be centered on how we think about making a bypass engineering device scalable. So the issue we ran into was that it was not scalable, right? So now I've sort of like run out of all of my engineering and neuroscience, and here becomes our journey through different fields to try to solve this problem. So at the time, I started wrestling with this question of, well, what is scalable? What sort of interventions have scaled? And I thought about two things that have scaled. The first one is antibiotics, and the second one is vaccines, right? And conceptually, in principle, what antibiotics do is that they target things that are not self to make self more sustainable or resilient in an environment, right? And as I wrestled with this idea, I said, well, at our core, neuropsychiatric illnesses are self, right? So that sort of concept of an antibiotic doesn't make sense. Now, on the other hand, what a vaccine does is it targets self to make self more resilient to things that happen in the environment. And I sort of like really love this concept of figuring out how to target self and make self more resilient to things in the environment. So we came up with this concept of, well, what if we could make a vaccine for mental illness? And we sort of sketched this down, which made no sense at all at the time. But I was invited to join the working group for the Brain Initiative. They were in their second half now. The goal of the Brain Initiative was to understand the human brain such that you could treat diseases like depression and bipolar disorder. And I learned about two technologies that they were working on, like sort of envisioned as these transformative projects for the Brain Initiative. The first one was the human brain cell atlas, right? And so they were going to take the human brain and its billions of cells, and they were going to sequence them and find the genetic sequence, sort of like gene expression sequence for each of these brain cells so you can target it, right? And these are sort of the same sort of patterns we use to target, brain cells in mice in the lab. Could you learn these about humans? So that was the first technology. And the second technology, these were technologies done by my colleague Viviana Gradnauer, Caltech, and others, are AAVs, which cross the blood-brain barrier, right? So in other words, you can give viruses, like the type of viruses we use to express channel rhodopsin in the brain. They'll cross the blood-brain barrier. And so if you paired a system like this with a human brain cell atlas, which is sort of your GPS system, you could deliver proteins to specific cells in the brain, right? And once I learned about these transformative technologies that the field was working on creating, I realized that the problem I actually had to solve was take this computerized closed-loop brain stimulation device and turn it into proteins. Because if I could turn it into proteins, we could package it in AAVs and deliver it to brain cell pairs of interest. So that became the challenge that we needed to solve in order to move something like this forward. So it turns out this wasn't far-fetched. I, as I said, went to medical school. And so I spent a lot of time thinking about ways that brain cells and other cell types transfer information from one place to another. So it turns out in the heart, one of the systems used to transfer electricity in the heart are through what we call electrical synapses, right? And electrical synapses, they're basically channels that connect two brain cells. They're also called gap junctions. And they allow ions to flow from one cell to another. It's why your heart is so synchronized, right? Because it can move electricity from one cell to another. And it turns out what it's actually doing is reading one cell and playing it in another, right? It's transferring electrical ions between one another. And this is done through a set of proteins called connexins. 6 connexins come together to form a hemichannel. 2 hemichannels come together to form a gap junction. And these gap junctions, they move ions back and forth. They're expressed in most human tissues. There are about 20 forms of them. And because the ions can move back and forth, they're usually bidirectional. And as we started looking through the literature, we found this really interesting use of connexin proteins. So I'll come back to here again a little bit later in the talk. But people had used C. elegans. C. elegans are worms. So we're, again, in a totally another discipline space. And C. elegans don't have these connexin proteins. They have another type called the nexins. But what that means is that you can put these connexins, what they showed, you could put these connexins in specific cells in a C. elegans, and it basically created a bypass in the C. elegans, right? It moved electrical activity from one cell to the next. It changed the physiology of the C. elegans, and it also changed the C. elegans' behavior. So you could basically rewire or create a bypass in C. elegans. So the problem with doing this in humans, let alone other mammals, is that the C. elegans basically has one cell of each cell type, right? That is not the case with humans. So imagine we wanted to put these gap junctions back into that sort of brain cell example I showed you. The gap junctions are the pink squares here connecting the two brain cells, allowing ions to flow, ions of electricity, between two brain cells to create our bypass. So let's say this is what we were trying to achieve. The first problem you would run into is because we have many brain cell types many cells of each brain cell type, you'd also get the gap junctions expressed there, right? And so as you have this signal upstream, the signal upstream would scramble because it starts sending current sideways instead of up and down. That's problem number one. Problem number two is information, as I said, these gap junctions are pores, so information can flow, electricity can flow in both directions. So you'd sort of get scrambling like this also, right? It's like when you're trying to talk to somebody and they're yelling at you, this is essentially what you would get. But, I like to give things a try in the lab. So I said, well, what if we make these gap junctions and we just try them out and put them in mice and see what happens? So we made these gap junctions. We put them into the hippocampus of mice. And we saw this, right? I'll never forget when my grad student, we were hoping the animals might like spin around or lose spatial navigation. That's a survival curve. And so a couple of days, we injected them at 2 doses. The mean dose is in the middle. Like 4 or 5 days later, the animals started to die, right? And my graduate student said, oh, they don't really have a phenotype. I said, there's no clear phenotype in the world, particularly when you're thinking about translation. There's nothing more important than your animals don't survive. It's a very clear phenotype. And the animals, their hippocampus basically became hyperconnected. They seized and they died, right? So it's the problem that I'm showing you on the left. Information's moving forward, backwards, and sideways. So they had a very clear phenotype. So the issue is that connexins are essentially like stickers, right? Like they stick together, they come between two cells, they let electrical information flow. So we asked the question, what if we changed what we were trying to do conceptually and we turned them into magnets, right? So instead of functioning like stickers, now they're magnets. There's a positive end and there's a negative end. And I apologize to anyone who's colorblind. These colors sort of are meaningful for the rest of the talk. But there's a green connexin and a red connexin. Imagine that one's a positive end of the magnet, one's a negative end of the magnet. And now when we express them, the positive and the positive repel, right? So even though it's at both presynaptic cell types, the positive and positive repel, but the positive and the negative can come together, right? And just for kicks, what if we can magically invent it so current tends to flow in one direction over another? And so this was conceptually what we were trying to achieve. And I pulled together what I like to call my dream team. And I call them my dream team because they were a group of undergrads, and they had no idea that this couldn't work. And so they start off with this magical belief that the answer's out there. And so they searched the entire literature. They searched through connexin proteins. They searched through other versions of gap junctions, panexin and nexin proteins. And they came up on this really cool pair, which is in perchfish. It's in goldfish also. And the connexins here, they're labeled by their molecular weight. So connexin 34.7 is 34.7 kilodaltons, and connexin 35. And this pair had this really interesting set of features. First of all, it's 2 different connexins that can come together, right? So it's sort of like maybe it's going to get us our green and our red. They tend to be located presynaptically and postsynaptically in nature, so that's really cool. They tend to rectify, which means current tends to flow more in one direction than another. And wonderfully enough, they tend to conduct current at exactly the physiological range you would want for human neurons, right? So we selected this pair, and we decided to move forward with it. The problem with this pair is that it does what we want it to do. In other words, it works like a magnet, but it also works like stickers, right? So the 34.7 will dock with 34.7, and the 35 will dock with 35, right? And so we've now gone a little bit further. We've touched C. elegans a little bit. But I suddenly realized that all those biochemistry classes I'd failed in medical school were extremely important. So it turns out the docking is at least in part controlled by residues on the extracellular loops of these connexin proteins. All of those words, I had no idea what it meant when we started down this adventure. But what we realized we could do was we could mutate the residues, right? And if we mutate the residues, maybe we can figure out which ones are positives, turn them into negatives, negatives and positives in the right combination, and essentially create a pair where they don't function like stickers anymore, but they continue to function like magnets. So that's what we're going to try to achieve. And I'm going to fast forward through the next slide because this was like years of work. But the way we solved it was we came up with a novel screening assay in the lab. I hired a biochemist in the lab to come up with a way of rapidly screening the interaction between different connection protein pairs. I had a computer science major join my lab, and he created some computational models. Neither of them could solve the problem on their own, but when I finally got them to talk to each other, they integrated their two systems together. It created a way of rapidly iterating through an in vitro screen and a computational model, and we're able to sort of crack the code on how the residues interact to create the binding between connection proteins. And it looks like this. There are essentially 4 really important residues, and the second one from the top on each side interacts with 3 on the other side, essentially forming like a connection mechanism. And so what we realized we could do was basically systematically mutate them to get the connection working just the way we want. And so 34.7, we made almost all positive. 35, we made almost all negative. It turns out that magnet concept actually mapped onto biology. And we were ultimately able to identify a pair with our features of interest. Here's what it looks like electrophysiology. We express this. They're proteins, so we can express it in hex cells. And then we were able to dual patch, so we're basically connecting a pipette to both, and we can measure the flow of electricity. And when we do this, we found that we can send electricity between one cell and the other. So these channels were conducting electricity, right? And we were really excited about that because it means even though we mutate them, they could still form a functional gap junction. And when we express pairs, so one cell has 34.7, one has 35. When we express 34.7 in both, I'll call like the lock and the lock. When we express the lock and the lock, no current passed between the cells. And when we express the key and the key, no current passed between cells. So we have it working like a magnet, and it doesn't work like a sticker, all right? So we reached out to colleagues. We sort of mentioned the C. elegans literature. We don't do C. elegans in the work in the lab. But I had a dear friend, Daniel Colon Ramos at Yale, and his lab does work in C. elegans. And I said, look, we've got these new cool proteins. Would you mind testing them out in C. elegans to see if they can actually change behavior and physiology in a living, intact system? So we sent them these proteins. And the C. elegans is nice for multiple reasons. One, as I said, they have one cell of each cell type, right? But what's also cool is neuroscientists have worked really hard to figure out the behaviors that are linked to each of these cells of each cell type. So there's a set of behaviors that are sort of mapped onto cell types in the C. elegans. They're clear, so you'll understand and appreciate why they're clear in a few slides. And then they have a total of 302 neurons, so less to deal with than if we're talking about a mice, which has tens of millions, or a human, which has billions, 100 billions. Okay, so here's the assay that we're going to use in C. elegans, and I'll walk you through this quite quickly. So I like to say C. elegans are like graduate students. Wherever you feed them, they will return. So this behavior is controlled, at least in part, by 2 neurons. There's AFD, which you can see it's the large green neuron over there on the top. And then there's AIY. AFD is a sensory neuron, right? And in this case, it senses temperature. So the warmer an arena the C. elegans is in, the more that neuron fires. So if you feed C. elegans at warm temperatures, and you test the C. elegans, it'll go back to warm temperatures. If you feed C. elegans at cold temperatures, it'll then prefer cold temperatures later because that's where it found food. So in the case of feeding an animal a C. elegans at cold temperatures, the sensory neuron responds to temperature because it's a sensory neuron. So underneath the big green neuron, you can see a trace in which we're increasing temperature. So we're ramping temperature up. And I'm showing you across 60 or so animals what happens to that neuron as we increase temperature. And you can see it sort of gets more red as you slide to the right because it's responding to temperature. So the neuron fires with temperature. It's a sensory neuron. But when you feed animals in cold, the connection between AFD on the left and AIY on the right is weak. And so the second cell, because the connection is weak, the cell doesn't fire more when you feed it at cold temperatures. And if you test the animals out, they prefer cold. But in the case where you feed the animals at warm temperatures, now the connection between those 2 neurons is strong. It is changed. It is potentiated. So, again, when you increase temperature, you see that the presynaptic neuron, or AFD, increases, but now the postsynaptic neuron starts firing more as well because the connection between these two is strong. And the animals prefer warm. So you can measure how strongly these two neurons are connected by two features. One, does the animal prefer cold? Does the animal prefer warm? And then secondly, how much calcium rise is there as you increase temperature? So it's a really easy way of measuring the connection between these two cells. So that's what we're going to do experimentally. We're going to put the lock in one cell and the lock in the other cell. Or we're going to put the key in one cell and the key in the other cell, right? And then we're going to measure calcium change and do the animals want warm or cold. So when we do this, I'm showing you the lock in the middle and the key on the right. You can see that there's no postsynaptic calcium rise, right? So in other words, we're not strengthening the connection between these two cells when we do lock or key. But when we put the lock and the key in, so the lock in the first cell and the key in the second cell, all of a sudden you can now see a postsynaptic calcium rise, as if we have strengthened the connection between these two cells, even though the animal's never, it's never gotten food at a warm temperature. So we are changing the wiring between these two cells. Now we're going to look at what goes on behaviorally. In the wild-type animals, as I said, they prefer cold because you fed them at cold. The animals with the lock and the lock prefer cold. The animals with the key and the key prefer cold. But if we look at the animals where we put the lock and the key in, all of a sudden, they prefer warm temperatures. So you've changed the physiology and you've changed the behavior, essentially creating a synaptic bypass that works in C. elegans. All right. And then, just for kicks, because my uncle shamed me with this question, we tested this against human connexin proteins that are in the human brain as well. Because this time we'd learned that we'd better think about long-range translation early on. And we found that these proteins that we created did not dock and interact with connexin 36, which is the human version, or connexin 43. We've since tested this against other ones as well. So it doesn't interact with them by docking. Okay, so now we're really excited, right? Like we've got this thing, we're playing around with it, it seems to be working in C. elegans. And I'm going to take you all the way around back to the beginning of my talk again. So if you remember, I put up this diagram about these genome-wide association studies and showed that a lot of the sort of gene alterations in humans were mapping onto one of a lot of different synaptic proteins, right? And so I'm going to spend the rest of the talk focusing in on one of these proteins. It's called SAPAP. And you could sort of see it. I wish I could get the pointer to work up there. But for all of you at home, you get to enjoy me doing the pointer on here. It's that little sort of green link over there that's holding up the AMPA receptors. And people had made a mouse model where they'd remove this protein. So they'd remove SAPAP3. And the work was first published in 2007. And when this got published, there's this really interesting phenotype in the mice, right? So as they aged, as they sort of passed through what is mouse adolescence, people started noticing that these animals started grooming a lot. Like they had a lot of repetitive behavior, and they groomed, and they groomed, and they groomed. And they were grooming about twice as much as a wild-type mice. Sometimes, some of them like 4 or 5 times as much as normal wild-type mice. But they groomed, and they groomed, and they groomed. And the consequences of that grooming could be quite severe. So I'm showing you a wild-type mouse on the top, and on the bottom, I'm showing you a SAPAP mouse. So a mouse that has sort of lost this protein, and it grooms, and it basically can groom until it has these huge facial lesions because it's grooming so much. And I'm just going to quantify that for you here. You can see the lesion size. This is the size of the lesion on both sides of its face averaged together. So 0 means no lesions. 5 means it has 2 5-millimeter lesions on both sides of its face. So you just don't see this at all in the wild-type animals. And in the knockouts, you see these lesions, and most of the animals have it, like 85% of the animals. By the end of this, we're tracking them for about 10 weeks. 85% of the animals by the end of 10 weeks. So 25 weeks of age, we're tracking them. From 15 to 25, they have lesions. Okay, so what's really interesting about this is that multiple labs have tried to map this behavior onto circuits in the brain, right? And they sort of implicated 2 brain regions that are particularly important for this. One is orbital frontal cortex, and then the other is the striatum. It's sort of like the central part of striatum. And the way this circuit is set up, I have a diagram here. You have cells in orbital frontal cortex that are sending projections into striatum, and this is sort of unidirectional, right? Like there's a circuit that gets information back around, but this is unidirectional. And people have used that tool, optogenetics, to do a set of experiments. Like people have stimulated the terminals of the OFC neurons in striatum, and it reduces grooming. People have also done a set of experiments where they target the PV neurons, so they set up interneurons in striatum, and they stimulate that, and that reduces grooming as well, right? And so it sort of argues that there's probably a circuit between orbital frontal cortex and striatum, but particularly the circuit in the middle, not the one on the left or the right, that's important, right? But there haven't really been great neuroscience tools where you can stimulate like part of that, but not the other parts, right? It's not selective. You sort of have to be able to hit the presynaptic neuron and only the postsynaptic neuron for this to work. So our idea was, well, we sort of created this like lock and key gap junction tool. Can we just test this out on that circuit and see what it'll do? So what we're going to do is put the lock in the orbital frontal cortex, and then we're going to put the key into one of those three cell types in striatum, either the D1 expressing neurons, the D2 expressing neurons, or the PV neurons. And then we're just sort of going to see what it does. Can we rewire the circuit and change behavior? We're going to create a series of viral vectors. I'm going to show you this and walk you through this on the next slide. But we're going to have a series of viral vectors, and we're going to tag green. So I warned you that you'd see green and red a lot. Our 34.7 is going to be tagged to the green. Our 35, which is the key, is going to be tagged to the red. And then we're going to have a series of controls, like GFP, which is just green protein, RFP, or M-cherry, which is just red. And then we're going to target these cell types. And I'm just showing you an image on the right. There's a picture in striatum. It's a confocal image. And you can see the green is sort of the axons coming in and the terminals coming in to striatum. And then the red, in this case, is a PV neuron. And on the bottom picture on the right, you could sort of see that like there's like a yellow spot in there that's like the green and red, and it's sort of yellow. It means that these two colors are sometimes like really coming close together, right? Sort of arguing that maybe we're creating a synapse there. So here are our experimental groups that we're going to test. We're going to start off with a strain of mice, these sap-up mice. We've crossed it to mice that will allow us to target PV neurons. So it's a PV Cre mouse, right? And so this will allow us to target all the cells that express Cre with a virus. And so we're going to put the lock, or 34.7, in the frontal cortex, and the key into striatum. Our control groups are going to be only color, only green, so fluorophore and OFC and striatum. That's on the top. The next, the lock is going to be OFC. The key is going to be specifically in the cells in striatum. The next one, the lock is going to be in OFC. The key is going to be everywhere in striatum, so it's going to target all cell types. The third one is the lock in OFC, color in striatum. Color in OFC, lock key in striatum. And on the bottom, we're going to target everything but the cells we want to target. So we're just going to target everything except for those cells, right? And I'm going to show you on the next slide what happens with grooming. This is all one experiment, but I've sort of split it into 3 panels so you can keep your eye on the data and how these animals look. Okay, so I'm showing you grooming and the amount of time the animals spent grooming. We put the virus in at 8 weeks. We measure their behavior 5 weeks later, 10 weeks later, and 15 weeks later, right? So these are sort of chronically expressed proteins. And on the case on the left, what you are seeing is in the light gray, you see the animals that only have colors in there. No proteins, just control colors. And their grooming is high, as we would expect. But in the animals that we've put the lock and the key specifically into those individual cells, it reduces grooming across the entire course of the experiment, right? So we can, by putting into these cell types, reduce the behavior. Then we're going to look at our control groups. In the middle, you see the group where we target OFC, so the lock and OFC, and the key is everywhere in striatum. So not just your cells of interest, but everywhere. That's the blue group, right? So if we target all the cell types in striatum, the grooming increases over time, right? So it's only the case that we've hit the individual cells, not when we hit all cells. And then in the purple, we have a case where we hit everything except for the PV neurons in striatum and OFC. So we're forming the connections at the wrong place, and the grooming increases over time, right? So it's only when you hit the cell type pair. And then all the way on the right, you have only the lock in striatum, or only the lock in cortex, only the key in striatum, so only half of the protein, and again, their grooming increases over time. So it's only if you put it at the right connection and bypass the right place. here's what the animals look like in terms of their facial lesions. I'm going to walk you through the groups again from top to bottom. So only colored proteins, the animals develop facial lesions. It's actually most of them. It's like 90%. So that is the control group. If you put the lock and the key in, 2/3 of the animals never develop any lesion whatsoever, right? So not only decrease their grooming, they develop no lesion whatsoever. If you put the key, the locking cortex, put the key in all the cells, so you're not being selective, you're targeting all the cells, those animals develop lesions also, right? And in fact, the majority of them develop lesions. If you put only the lock in, this is now the 4th from the top, those animals develop lesions. If you only put the key in, those animals get lesions. And if you put the lock in and the key in the wrong place, right, not your cell type of interest, those animals also develop lesions, right? So you have to hit the specific synapse. You have to bypass the right thing. If you bypass anything else, you still have a problem. Okay, so I'll take you back to this map. We just did a series of control experiments as well, in which we target the D1 neurons or the D2 neurons. We either use a D1 Cree mouse or an A2A. It's a receptor that's very sort of overlapping in striatum with D2 neurons. And when we do that, we find that doesn't help, right? So those animals all develop problems grooming, and they develop lesions as well. So you have to hit the cell type pair of interest, or it doesn't work. You have to bypass the synapse of choice. Of course, we're also excited because it turns out you could bypass the synapse and keep the animals from essentially grooming their face off if you hit the right thing. Okay, so we're sort of left here, and I'll spend the last sort of 2 or 3 slides for all the physiologists in the room, because at some point I got to come back to my own discipline, right? And so we went through, and we wanted to record what was happening with the code in this brain area. And I apologize to all the non-physiologists in here. This will get a little bit nuanced. I just want to show my neurophysiology creds when I'm here. But we implanted a type of recording electrode that allows us to record lots of neural activity, right? So we're going to do very high-density neural recording activity from striatum. And then we're also going to get that same sort of oscillatory information from orbital frontal cortex all at the same time in a mouse. And so we're going to collect lots of cells. We're going to go to about 302 single neurons, about 900 total neurons. And we're going to record oscillations from orbital frontal cortex in the same cell. And then we're going to split the neurons into 2 groups. For all of those who study striatum, you'll sort of digest this really quickly. We have neurons on the right, which sort of have narrow waveforms. These are going to be the cells that are likely your PV neurons. And then we have neurons on the left with wider waveforms, and these are going to be what we call medium spiny neurons or striatal projection neurons. And so those are your 2 classes. And then we're just going to ask, well, how is the activity of these cells different under normal conditions, and then when we sort of do this bypass? And so on the left, I'm just showing you a normal animal and the knockout animal, right? And so when we look in striatum between the normal animal and the knockout, there's no bypass here. There's basically the striatal projection neurons, or medium spiny neurons, fire less, right? And so there's a decrease in their firing rate. But when we look at the narrow spiking units, even though there tend to be less of them, right? There's no statistical difference in that distribution, right? Neither of them is normally distributed. But in terms of the mean firing rate, there's no difference, right? And we've got a lot of cells there. So we were actually surprised by this because we expected a lower firing rate of these neurons, and we thought that's what would be happening experimentally. We then moved on to study, well, what's going on in these animals when we do the bypass? So on the left, you see the control group. On the right hand, for each of these 2 plots, you see what we call links. That's our bypass. When we put this sort of bypass in there, the firing rate of the medium spiny neurons got even lower, right? So you sort of have the disease case, the firing rate is lower. And then we sort of fix it, and the firing rate gets even lower, right? Which is super confusing to us. And then we looked at the narrow spiking units, what we thought was the PV neurons. And even though it tended to increase the firing rate, there just weren't a lot of PV neurons there. There was just we're doing deep recordings, and we're barely getting any PV neurons or things that have waveforms there. So we were actually struck by this. So we just quantified, well, what percentage of neuron types do we have? And that's where it got really interesting. So on the left, you see our normal animals, the SAPAP plus, and the knockout animals, the SAPAP negative. And what we could clearly see when we quantified this across animals is that there was just less neurospiking units, less of these what we thought were PV neurons in the mutant case. And then when we looked at the mutant animals with and without the Lynx protein, what we could see was that we increased the proportion of PV-expressing neurons or neurospiking units. So you can see the red, the slice of the pie gets smaller, and then on the right, the slice of the pie gets bigger. Interestingly enough, on the right, the total number of PV neurons was way less. We think that has a lot to do with the Cree line, which just decreased the functionality of the PV neurons there. But we think what's happening is there's essentially a silencing. In the disease case, the neurons just basically go silent, right? So if you're doing something like calcium imaging, you can see them. If you're doing something like electrical recordings, they just sort of drop off. Your ability to assess them, you can't see them because they're not firing, right? So there's a silencing of these neurons. And when we bypass them, it recovers these silenced neurons. Okay, I'm gonna just fast forward through the last two slides and sort of give you the take-home of what we think is actually going on. So in the end, we take these neurons that are firing and we ask, how well are they connected to OFC? So we can quantify what is the firing relationship of the cells in striatum to the real-time oscillatory information in OFC. And what we see, I'm just showing you on the left, is that when you look at the knockout animals, their cells basically show they fire in a way that has less of a relationship to OFC. So it looks like there's sort of this disconnection in the circuit between the two because the cells don't have a synchronous timing with OFC. When we go to the right-hand side, we see what happens to the mutants. When we restore this connection, we sort of restore the firing relationship between them. Now, the cells in striatum are firing with a better relationship with the connectivity in OFC. And so we think ultimately what we've got, and I'll just sort of summarize this, is that our goal, we set out to basically bypass the synapse, right? And we thought that in the case of a mammalian species, in this case a mouse, we could target a mouse in which there's a sort of a genetically induced dysfunction in the synapse. And so we've got the synapse, we've got the brake, and we want to know if we can bypass the brake. We came up with a solution of basically generating bypass protein channels, which we turned into magnets. We tested these in C. elegans. They looked like they worked. We went into the disease model in which the mice repetitively groom until their face is basically scratched off. We identified a circuit that had been implicated, a specific connection of interest, put the bypass in there, and it restored both the physiology of the circuit and normalized behavior and got rid of the pathological outcomes. So we're not there yet. This obviously isn't human translation. The story isn't over. We're still doing a lot of work to get there. But I just wanted to take the last minute and a half to talk about the people that did the work. And I always end my talks with a slide that looks like this, because I think what is so fundamental to what we're trying to achieve is to get the smartest, most talented people working on the hardest problems. And for me, when I was given the opportunity to come here to NIH, I just wanted to take the opportunity to thank NIH for all the work that it does in removing barriers to the smartest people working on the hardest problems. So that's me in the upper left-hand corner. Believe it or not, I own more than one outfit. Next to me is Liz. She's a first-generation college student who's a postdoc in the lab who came up with the cellular screening mechanism. She's now in a junior faculty position, started her lab at Carnegie Mellon. Next to him is Ryan. Ryan grew up in San Francisco, went to Montessori schools. I met him as an undergrad in my class. He was super weird. I thought he wasn't paying attention. He got more than most of the people in the class. He joined my lab. He since did his master's at Carnegie Mellon. Elise Adamson was an undergraduate major at the University of Maryland, Baltimore County. Came in, did her PhD in biomedical engineering in my lab, and now works for Bain Capital as a consultant. Tatiana Rodriguez was an undergrad at the University of Maryland, Baltimore County. Grew up in the Appalachian Mountains in Maryland, super rural area. Came to my lab as a technician for 2 years, is now doing her PhD at NYU in environmental engineering. Steve McGue grew up in Maine, did his PhD work at Penn in the area of addiction, and came down to my lab. He helps me run it. Hannah did most of her work on the SAP model, spent an incredible amount of time growing up in Dubai, is a technician in my lab, and is going off to Columbia to do her master's in psychology. Dalton Hughes was an undergrad at the University of Maryland, Baltimore County. He's right there in the middle. He did his MD, PhD at Duke, and is now working for a pharmaceutical company and a biotech company, thinking about how to create new translational tools to treat people with mental illness. Danielle Colon-Ramos, lab at Yale, my colleague. Catherine Walder did her PhD in biochemistry at Duke before joining my lab. Catherine had 2 kids during the pandemic, and it was when I realized how important it was to think about childcare in supporting young scientists as they were sort of doing their important work. Next to Nicole Kalakis, she's an MD, PhD at Duke in neurology. Eli Wisdom, who is an undergrad, who worked in Daniel Colon-Ramos' lab, is now an MD, PhD student at Oregon Health Sciences. Hannah, who was an undergrad in my lab, she was part of the dream team that sort of cracked the code on finding the right protein, is now a cardiology fellow at Duke. Gwen, who was an undergrad at the University of Maryland, Baltimore County, finished with her PhD in neurobiology at Duke, and is now working for a foundation. Ninad Bursak was the first person I told about this idea. He was faculty in biomedical engineering, working in cardiology, and family's originally from Serbia, came on board to help with this project. Kirill Chesnov was a PhD student in my lab. He's the one that did a lot of the computational modeling, is now a postdoc at Stanford. Half his family migrated here from Russia, the other half from Ukraine. And finally, Rainbow Holtman, who I always like to end with. She was the one that did the original sort of finding the right mutations in this code of the connection proteins. Rainbow developed debilitating migraines when she was a graduate student at Duke, so much so that she couldn't look at a computer. She figured out how to navigate that disability, joined my lab as a postdoc. She is now running her own independent lab at Iowa, and she was awarded NIH New Innovator Award to study the neurophysiology of migraines. So thank you. I'm happy to take any questions.
And we do have time for questions. The CME code, for people who might not have caught it in the beginning, 57693. Looks like we have a question coming. No, we're not. Oh, you got one right here.
Yeah, that was an absolutely wonderful lecture for many, many reasons. But I'm going to be very specific in terms of what actually your thoughts are on this, in this pathway that you are targeting, that is eliciting this compulsive behavior from OFC to striatum, which is exactly the same one that we're seeing in compulsive drug taking. So have you added giving drugs to these animals? Do they escalate faster?
So we haven't done this. This data is about 3 months old. I do have a graduate student who's actually supported by you also. Thank you very much. Who is looking at both fentanyl consumption and cocaine consumption and thinking about how to rewire circuitry to change that behavior as well. We don't have an answer yet.
And my question to a certain extent was, because I suspect that there's going to be some specificity for behavior, because you're speaking these animals are compulsively doing things, but they are not compulsively doing other things, I assume. I don't know.
Yeah, I don't know if anybody has looked at these specific animals in that instance, right? I'm not sure. Yep.
Kaf, this was a great talk, man. I super appreciate seeing it, especially the physiology. I have a question to you about that.
Yeah.
Have you guys sort of the function of the LNC is going to depend on where it forms its connections, the dendrites, the cell body, and have you guys looked at where it forms the connections and what it does to the sort of the physiology of the cell?
Yeah, no, if only I had a collaborator who studied synaptic physiology. I'd be interested in, So we haven't done that, right? Like for us, there's so much to explore with a tool like this and so much to learn. And a lot of it just falls out of the range of what my lab does. So we are happy to share. We've shared the tool with a lot of labs who are playing around with this. But if that was something you were interested in tackling, we will walk the stuff. I will mail it on ice, but we will get it up here to you.
Cool, man. Appreciate it.
Okay. Kind of a double question here. First, the person's curious about the timeline. How long did this take? You said a long time, but...
Sorry, are you referring to the mice or the entire scope of the project and the vision?
I think the scope, how long it took to get to the mice, because the follow-up question is, what's next vis-a-vis animal?
Great. Yeah, okay, great. So it was 2014 14 or 15 that we knew what we were doing wasn't going to be scalable. It was 2018 when I started on the Brain Initiative panel, right? So that sort of anchors the middle of the talk. Everything up to that point was probably 2005 to like 2013, 14, right? Yeah, so long runway.
Then after the mice, do you have, what's they're curious, the next animal model you might approach?
Yeah. So I won't introduce that now because we'll be here for the next hour. It's a totally another talk that requires a lot of background information. So I won't answer that, but if you ask me at the reception or if the person's here, I'm happy to get into that. But we are already testing this out on human organoids as well to make sure that like human brain cells can tolerate the protein and that it can change physiology there as well.
Excellent. Any other questions? One more. Hey,
Very nice talk. I just had a question about the stress. So the mouse is hanging upside down. That's the anxiety component. And so you did it for one day and then another repeat day. But who's to say that third day things wouldn't be different, or the fourth day or the fifth day?
Yeah, so people have tested that, right? And the animal sort of, its behavior keeps looking more and more and more like a more and more stressed mouse, right? When people have checked it like 5 or 6 days in a row, right? There are other sort of more robust ways, that sort of ethologically anchored ways of stressing mice. We just like the simplicity of this because we could, record electrical activity from the mouse's brain while it was hanging upside down to identify the circuitry, right? For testing, if ultimately we're now testing this in stress models or in other types of stress paradigms, whether it's something like chronic unpredictable stress or chronic social defeat stress. But the idea is you should be able to target the same circuit if it is a stress-resilient circuit, and you should have translational relevance there.
Couldn't the mouse get used to be hanging upside down?
Yeah, so this is why I don't want to make claims about whether the mouse's behavior is good or not, right? Because there are lots of ways an animal can adapt to an environment. And some might argue that it makes a lot of sense to stop fighting so hard about a situation that you can't get out of. Save your energy, save time. At the end of 5 minutes, the investigator is going to take you down, right? So I don't want to make claims about whether it's good or bad. And it's certainly why I don't believe it's depression, right? But it is stressful. And the stress does cause a change in behavior, right? And I think we can use that as a way of identifying circuitry.
Okay. So if there's no more questions, let's thank the speaker one more time. There is a reception, as Kaf had mentioned, right outside.